Welcome to ANA Investigates, a monthly podcast featuring the latest in neurology and neuroscience brought to you by the American Neurological Association. I'm your host, Adeline Goss. Less than a year. That's how long it took between when the novel coronavirus emerged and when a vaccine was available to prevent it. This incredible speed of vaccine development exemplifies scientific innovation in a time of crisis. There's increasing vaccine acceptance among the public, but questions about target populations, mechanisms, and side effects remain. Neurological complications have been documented with other vaccines, and there have been some possible complications observed with the vaccines for COVID-19. Here at ANA Investigates, we figured we'd explore this topic by going to the source. My guest today is Dr. Avindra or Avi Nath. Dr. Nath is a physician scientist who specializes in neuroimmunology and infections of the nervous system. He's the intramural clinical director of the National Institute of Neurological Disorders and Stroke at the National Institutes of Health. Welcome, Dr. Nath.
Thank you very much, Dr. Goss. It's a real pleasure to be on the podcast.
Dr. Nath, I wonder if you could start us off by walking us through the approved COVID-19 vaccines and candidate vaccines. Which vaccines are out there, and what are their mechanisms of action?
So there are at least close to 100 different vaccines that are in various phases of preclinical or clinical studies. And the approved ones are a small fraction, but you can largely divide them into three major groups. So the DNA-based vaccines, RNA-based vaccines, and the protein-based vaccines. Then the last one is also an inactivated virus. So the DNA-based vaccines are, for example, the AstraZeneca vaccine, and that is, it's made in an adeno-associated viral vector. So just the spike protein is expressed in most of these vaccines. The Russian vaccine is also an adeno-associated viral vector, but it's a different type of an adeno-associated virus. Then you have the RNA vaccines. The RNA vaccines are the Moderna and the Pfizer. And they are also based on the spike protein. The RNA encodes just the spike protein alone, which is the coat of the virus. But there's a slight difference between the sequence that is present in the RNA vaccines compared to the AstraZeneca vaccine. The Moderna and the Pfizer vaccines mutated two amino acids that allows for much greater immunogenicity compared to the AstraZeneca vaccine. And that's why you see a difference in efficacy as well. While the RNA-based vaccines are about 90% effective, the AstraZeneca is a little bit lower than that. And then you have the protein-based ones, and that's the Johnson & Johnson vaccine is based upon a protein that is derived again from the spike protein. And that is a single injection only. But again, the efficacy is a little bit lower, but yet they're very, very effective. And then lastly, you have the inactivated virus. So the inactivated virus is the Chinese and the Indian vaccines that they're using. They've grown viruses to large levels. They inactivate it chemically and then use that as a vaccine. That also requires 2 dosages.
Great, thank you. So what do we know about neurological complications of these new COVID vaccines?
So there's been a lot of interest in the possibility that there may be neurological complications associated with them. Early on, if you might remember, there were cases of Bell's palsy that were reported. And in fact, when the Pfizer went to the FDA, they reported those cases of Bell's palsy. But it turns out that the number of cases were no different than what they would have expected by chance alone. There are other patients who have complained of possibility of fasciculations in their hands, tingling sensations from the site of injection. There are people who have complained of brachial plexitis type symptoms, but by and large, most of these things are fairly rare, and it's hard to know whether they're greater than by chance alone or not. The American Academy of Neurology released a statement, and they thought that the incidence of neurological complications is not significant with the vaccines themselves. So even if there may be some, I think the most important thing is that it shouldn't dissuade people from taking the vaccine, because the consequences of the infection itself outweigh the potential risk that may be associated with any of the vaccines.
I think many neurologists were concerned when there was a report of transverse myelitis associated with the AstraZeneca trial. Can you tell us a little bit about, what wound up happening with that and whether we really think the vaccine was responsible for the case of transverse myelitis we all may have heard about?
Yeah, so that's a very important question. So there were two patients that were reported after the AstraZeneca vaccine was first initiated. Both of them were in the UK. And the first patient who developed symptoms suggestive of transverse myelitis turned out that she actually had multiple sclerosis, and it was an exacerbation of her disease. The second one, from the best that we know, this patient was originally thought to have transverse myelitis. However, this patient recovered fairly rapidly. So then they changed the diagnosis and called it a myelopathy of some sort. But we don't have the exact clinical details. I think all the data was presented to the FDA. It's not publicly available, but the FDA looked at it and they did not seem to be concerned. And certainly there haven't been other cases reported since then. So I don't think there's any major concern about the possibility of transverse myelitis from any of these vaccines.
Is there anything we know about the coronavirus that's concerning in terms of a vaccine's potential to produce an autoimmune response, or do we have some data that's reassuring against that?
So the two things. Number one is that those individuals who are infected with the virus, they are developing some immune-mediated phenomena. And so certainly with the full-blown infection, that seems to be somewhat of a concern. But if you just take the sequence of the spike protein and you blast it against the human genome, it doesn't come up with anything that has homology to it. So I think from a vaccine standpoint, it's highly unlikely that you're going to produce any kind of an autoimmune syndrome there.
It seems like one issue is that in any population, there's a background rate of neurological conditions like Guillain-Barré and facial nerve palsy, as you mentioned. And so just by chance alone, some people will develop these conditions in the weeks after receiving a vaccine. How do we establish whether a vaccine is actually responsible for that condition or not?
The data has to be generated through epidemiological surveys. And so there are two databases that exist currently to capture this kind of information. One is called the VAERS, which is the Vaccine Adverse Event Survey. And so anybody can go to that website and disclose whatever side effects they may have experienced. And so that data is collected. Now, nobody goes and confirms the accuracy of that data. So there's that flaw there. But nonetheless, it is still a rich database and is publicly available. So anybody can look at it and try to determine at least patterns of what you may be seeing with these vaccines. And it has data from all the other vaccines as well. So the opportunity is to compare it against other vaccines that have been administered to the populations. They've created a program whereby if there is any complication that occurs, that's settled by the government itself. So the manufacturers are often not responsible, and that's the incentive that they provide them to make these vaccines. The second database is called the FAERS database, which is FAERS. That is reported to the FDA. So that is reported usually by the company, the manufacturers themselves. And that is also publicly available. It's called MedWatch. So an application has to be filed and you can access that data. And so those two are the best ways of being able to capture very large databases to analyze them. Certainly one can do a prospective study in smaller populations if you want to, and follow people very carefully and determine if there is a true association or not.
I think one question that many of us clinicians are facing right now is that we care for patients with neurologic disease who are treated with immunosuppressive medications. Are these patients at higher risk of neurological complications from the COVID vaccines?
Actually, quite the alternative. So it's an interesting dilemma. And the thing is that most of the complications are immune mediated, right? So if they're immune mediated and you're on a medication that is suppressing your immune system, Your concern is not going to be the side effects. Your concern would be that are you going to get an adequate immune response from the vaccine to protect you or not? And so a lot of people are interested in this question. And I sit on a panel for the National Multiple Sclerosis Society where this topic was very heavily debated. And they just recently published some guidelines as to how one should dose the disease-modifying therapies in the context of getting the vaccine, and if the opportunity arises, to space them to whereby you have the least amount of effect. So that is the major concern there. I think the side effects of these things, if anything, there should be less in people on immune modulatory therapy.
And how about patients who have experienced a neurologic event that may have been associated with a previous non-COVID vaccine? Should they avoid the COVID vaccines?
So the only guideline that has come out is with regards to anaphylaxis. So if you have a history of anaphylaxis, either to a vaccine or for any other reason, then the chances of getting anaphylaxis go up. But beyond that, there should be no reason to fear the vaccine. I think even if you had some previous symptoms from a previous vaccine, I don't think there should be any reason. This is a totally different protein, so the immune reaction would be very different. And people worry about molecular mimicry and stuff, so those things are not going to be the same for each vaccine because the sequence is so different.
So as more people across the globe are vaccinated against COVID-19, where should researchers interested in this topic focus their attention?
So as neuroimmunologists, we are very interested in trying to see what kind of immune response they're developing to this vaccine. So I think that's one way to go. If you're interested in categorizing the side effects of it, then I think researchers need to collect large databases, do good, proper, controlled studies, prospectively collect data on these patients, and that could be very informative. And if you're a virologist, you want to see that, okay, is it really spike protein alone, or because one thing that one should be concerned about is all the mutations that are occurring with this virus, and are they going to be escape mutants? And so for us, we want to look and see whether we need to either incorporate other proteins in there, or if multiple different spike protein sequences should be included, and which ones should be there. As neurovirologists, we're also interested in looking at, I told you that when we blasted the sequence, we didn't find any homologies, but if there are new sequences arising, can that actually change? So as the virus is evolving, as neurologists, we want to be careful in trying to determine if there are new manifestations that may be occurring, or if the clinical syndrome may be evolving along with mutations in the virus.
Great. Dr. Nath, thank you so much for joining us today.
Oh, it was a real pleasure. Thank you for having me, Dr. Goss.
Again, that was Dr. Avindra Nath, the intramural clinical director of the National Institute of Neurological Disorders and Stroke at the National Institutes of Health. We'll have links to some of the articles he mentioned on our website. That's it for our show today. If you'd like to hear more episodes of ANA Investigates or submit your ideas for future podcasts, head to myana.org and click on the Education tab. For ANA Investigates, I'm Adeline Goss.
This has been an episode of ANA Investigates, the official podcast of the American Neurological Association. Thanks for joining us for this episode. If you're not already a member of the ANA, please visit myana.org to learn more about the benefits available to ANA members, including access to an array of educational programs and resources, like online CME courses, webinars, the ANA's journals, the Annals of Neurology and the Annals of Clinical and Translational Neurology, and more. Dr. Adeline Goss is our host. Our producers are Drs. Romer Giucaden, Adeline Goss, Michelle Johansson, Clifton Gooch, Danny Bega, Megan Ritchie, and Jim Sigler. This is your editor, Jen Hurley. Copyright, the American Neurological Association.